a beautiful microbiological influence corrosion session. So first, it is in the hybrid session, we will have more lecture style, but hopefully some interactive as well. Don't fall asleep. And in the afternoon, we do the labs, right? So that is really cool. So it's my pleasure to introduce the first speaker. Lisa Gieg is a professor here in University of Calgary in the Department of Biological Sciences. So yes. Okay.
Yes, thank you very much. Good morning or afternoon or evening. I know it's morning here in Calgary, but maybe evening somewhere else, people joining in. So myself, Lisa Gieg, I'm from University of Calgary. I'll tell you a little bit about myself in a moment. And Torben Skovhus is our actually really exotic speaker. He's come all the way from Denmark to kind of help with this workshop today. And so he will introduce a little bit about his background before we get started. So we're very happy. Thank you very much for inviting us to give you a day on microbiologically influenced corrosion. We often abbreviate this as MIC or MIC, so we'll probably use these terms a little bit interchangeably throughout the day. So I thought just to start, I'll give you a little bit of, let's see here. I have to move this. Yeah, just a little bit about me so you know kind of my background and where I'm coming from. So I was trained as a microbiologist, and I've always focused on problems of environmental microbiology. So I don't do a lot of medical stuff. Well, actually, no medical things. So I consider myself an environmental microbiologist. And a lot of my career has been focused on looking at how microbes impact the oil and gas industry. Okay, so whether or not they're involved in things like bioremediation of oil spills, or looking at microbial corrosion of oil and gas infrastructure. Being here in Alberta, we have a lot of, the oil sands industry, so we also do a lot of work in looking at how microbes can affect different parts of the oil sands industry. So I have been a professor here. I teach in the biological sciences department. I teach microbiology courses. So I've been a professor here since 2008. Just a little bit of background. Okay, so what we're going to be doing today, we're very excited. It's a beautiful rainy day outside. It's a great day to be indoors and learn about microbial corrosion. You won't have this desire to go outside, maybe. So this morning, we're going to give you a series of lectures. So Torben and I are going to kind of trade off our material. So this morning, I'm really going to talk, I guess, first thing about the basics of microbiology. So my understanding is that many students here are not microbiologists, right? Electrochemists or engineers. So we're going to have some really fundamental information about microbes, and then some really kind of introductory comments about how microorganisms can potentially, contribute or influence corrosion. And then Torben will come in and talk about corrosion management and MIC assessment. I'll be back to talk about some SOPs, so a little bit more detail on how we can look for microbes in different environments. And then Torben is also going to give you some failure analysis cases. And then in the afternoon, this is like really the fun part. So we have some really, what we think are really fun hands-on lab exercises. So you're going to get to clean corrosion coupons that have been incubating with microbes for at least a month. You're going to get to extract DNA, actually from kiwi, which will be really fun and very visual. And then we're also going to do ATP analysis. So if you don't know what any of this stuff is, that's what we're going to tell you about this morning. Okay. Okay, so I'm just going to, I think everybody here, of course, knows about corrosion. So we always kind of define it as an interaction between a material and its environment, resulting in the degradation of that material. We heard on Monday from Dr. Monica Hernandez about the high cost of corrosion. A big corrosion industry consortium called NACE estimated that corrosion costs about $2.5 trillion per year. So that's in damages, replacement, monitoring, prevention, et cetera. So it's a really, really huge amount of money is spent on this industry. Unfortunately, the Canadian Energy Pipeline Association doesn't exist anymore. But when I found this figure a few years ago, even just Canada's energy transmission pipeline industry alone spends all close to $3 billion Canadian to monitor and maintain pipelines. So it's very expensive. And then if you ever have a spill from a pipeline, it can cost upwards of at least $1 billion, right? So huge, huge, important and costly industry. Okay. So today we're here to talk about microbial corrosion or microbiologically influenced corrosion. And so where do microbes kind of fit into this system? So we can think about MIC as being a type of corrosion that's affected by the presence or activity of microorganisms. Okay, and when we think about microbial corrosion, we're also thinking about, 3 different components. So we're going to be talking a fair bit about the microbes themselves. So I call it microorganisms or microbes. So these are terms that I'll use interchangeably. But we also have to remember that that's just one piece of the puzzle when we're talking about corrosion. We also have to consider the medium, okay, the chemistry around where the microbes live, as well as the metal, right? Corrosion doesn't happen without the metal or the material. I think a lot of our content is focused on metals, but there certainly can be other types of materials that can potentially degrade or corrode. So you're going to see this diagram a fair bit to just remind you that, we are kind of focusing on this microorganisms part, but all of it is really important when we're talking about corrosion influenced by microorganisms. Okay, so I want to give you a really high-level overview, I think in 8 slides or less, all about microbes, okay? And so microorganisms, I guess one of the first really amazing things about them is they can live almost anywhere, right? So they can live in many, many diverse habitats, and I have a number of pictures kind of on the bottom here. You're actually going to be looking at ATP today from soil and from ponds. So kind of, those types of environments. Organisms like ruminant animals, their gut system is full of microorganisms that do amazing things for them. They live in oceans, they live in hydrothermal vents, they can live in the Arctic, in sea ice, they can live in deserts, and of course they can live on surfaces of things like pipelines and lead to corrosion. Okay, so they're incredibly diverse. When we talk about microorganisms, we're usually talking about bacteria and archaea. So these are kind of what we call prokaryotic organisms. They're kind of one branch or 2 branches of the tree of life. We're on the 3rd branch as humans. People also kind of clump viruses into the microorganism category, as well as fungi and algae. So microbes can live at a huge range of temperatures from about -5 to 121 degrees Celsius. They can live at huge ranges of pHs, and they can live at various salinities as well. So it really, that surrounding chemistry and environment can really dictate what types of organisms are in a certain environment. So they're incredibly diverse. So I want to talk a little bit about the molecules of life. And so I should also mention that we have all of these things in our cells. And of course, microbes do as well. But we're going to kind of revisit a few of these molecules later when we talk about how we can measure microbes. And you're again going to be looking at a few of the important biological molecules in the hands-on exercises this afternoon. So these are kind of the main types of structures. So we have proteins. Proteins are incredibly important. These are enzymes. So these are the sort of the types of biomolecules that transform reactions. They also important at transporting substances into and out of cells, and they give the cell its structure as well. Lipids are very important. They kind of have they surround microbial cells in a membrane, and they provide places where energy transformations can happen. Carbohydrates are types of glucose or sugars, hydrocarbons. They provide energy for cells. Nucleotides have various roles in metabolism. And the one we're going to talk about today is ATP. And so this is a molecule that's considered to be an energy currency of the cell. So cells will kind of they'll break down different carbon sources, and they'll capture the energy from those carbon sources as ATP. And then they'll use that ATP molecule to do all kinds of work for the cell. And then we have nucleic acids. So this is our DNA. So our DNA is essentially the blueprint of life. So we all have it. It kind of gives us all of our hereditary traits, and microbes have it as well. So we can use a lot of DNA-based techniques to identify microorganisms in samples. And then there's another molecule called RNA, and it functions in making our proteins. So it kind of links up to the proteins there. So just really quick overview of the different molecules of life. Just a little cartoon structure that I got from the internet showing a lot of the different parts of a microbial cell structure. So the cells are typically have a cytoplasm, okay? And one of the things about microorganisms is they're quite a bit different from us. Our cells have a lot of little membrane surrounded compartments, whereas microorganisms just have one big massive compartment in the middle that has all kinds of stuff in it, okay? So we call that the cytoplasm. We can kind of think about it as a gel-like substance made up of water and all the chemicals needed for growth. So carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. It's also surrounded by this cytoplasmic membrane. So that's composed of a lot of lipids and some proteins. And that actually is really important for the cell because it controls what can go in and out of a cell. Okay. And then we have other surface structures, that flagella, that's kind of this long skinny thing here, kind of helps it with motility. A lot of microbes have pili, so little proteinaceous appendages, and that actually plays quite a role in actually allowing some microbes to attach to surfaces that may ultimately lead to corrosion. And then they can also produce some slime, which is also important for biofilms, again, which might be contributing to corrosion. So, a lot of really important structures to understand also when we're trying to understand what they might be doing to surfaces of materials. We also have our DNA and our RNA. So our DNA is kind of our genetic blueprint. And I've put the kiwis up here because you're going to be extracting DNA from kiwis today. Not microbial cells, but actually, and the reason for that is you'll be able to actually see the DNA. Usually when we extract DNA from, let's say, water or soil, we actually can't really see it in our tubes, but we can actually see it today. So it's really exciting. I love it so much. So you'll be able to extract the DNA, okay? And that DNA, if we were to sequence it or figure out, what the composition of that DNA is, it'll give us those characteristics of that kiwi, right? So that's the genetic blueprint. And also really important is this molecule called the 16S ribosomal RNA. So it kind of functions, it's a very stable molecule. I'll go into it a little bit later in my second lecture. But we can actually use this molecule to help us identify what types of microorganisms are in samples. So we use an approach called 16S ribosomal RNA gene sequencing. And I will go into that a little bit later. But I just kind of wanted to introduce that molecule. And then just a little bit about this central dogma. So the way all cells work, including us, we have our genetic blueprint, our DNA. That gets translated transcribed into RNA, and then that RNA gets, translated into proteins. And again, those proteins are really important for all of our enzymatic functions. So it's kind of called the central dogma of biology. Okay. In the natural world, microbes can exist in a couple of different forms. So they can exist as single cells that are just kind of floating around, let's say. And so we call that planktonic, okay? So you might hear a little bit about planktonics. But most organisms, I think, prefer or can live in what we call biofilms, okay? And again, this is really important when we're thinking about corrosion. So biofilms are cells that are surface attached. We call them sessile cells, so they're attaching to a material. And most microbes are thought to actually exist in this form. And so if we take a look at, there's been a lot of different biofilm studies. A lot has been done in the medical field. For example, Pseudomonas aeruginosa is a type of microbe that colonizes people that have cystic fibrosis, so it colonizes their lungs. So there's been a lot of work that's been done on that type of microbe. We're not as, that's not as prevalent in the environment, but we know a lot of information about biofilms because of that organism. So if we take a look at the composition of biofilms, it'll have some of those different microorganisms. It's got something called extracellular polymeric materials, or EPS. That's kind of that slime that microbes can produce that contribute to a biofilm. There's going to be a lot of organic matter and inorganic debris as well, especially in these kind of natural looking biofilms. And the reason why a lot of microbes, let's say, we don't know if they like to live in biofilms, but often exist in biofilms is because they have a lot of advantages that biofilms confer. So promotes colonization, it ensures cell survival, and that's because they're kind of all grouped together in an this attached material, and they kind of protect each other. And that slime that they produce actually impedes chemical, like if you wanted to treat them with chemicals and get rid of that biofilm, it's really hard to do because the chemicals and that slime kind of prevent those chemicals from killing all of the microbes in the biofilm. So they offer really nice protection. Biofilms also ensure good nutrients. And as I mentioned, yeah, lots of different protection in a biofilm. So when we're thinking about corrosion by microorganisms, again, corrosion is a surface phenomenon. So we're looking and really most concerned about the microbes that kind of live on a surface in biofilms. Okay, so if we take any type of sample, what's really going to impact what types of microbes that you'll actually find in that system is the surrounding chemistry. So if we go back to this diagram and we see we've talked a little bit about microorganisms, that media, so that chemistry that's kind of surrounding them will help to influence what types of microbes will actually exist. And that chemistry will provide their food, right? The macro and micronutrients, the drink. So microbes need water, actually. They don't need a lot, but they do need water. It'll provide energy, so an electron donor. That could be organic or inorganic. And so that would be something like glucose or sugar, for example. And then, that surrounding chemistry will also kind of foster appropriate conditions for microbial growth. So, temperatures, pHs, whether it's aerobic, anaerobic, et cetera. And so there also has to be the absence of conditions that inhibit growth, okay? So even though I've mentioned that microbes are incredibly diverse, they can grow anywhere, there's going to be some harsh environments that might be full of chemicals that just won't allow microbes to grow. But what kinds of microbes will grow in a certain environment is really dependent on that surrounding chemistry. Okay, so I want to talk a little bit about, again, how remarkable microbes are because they can breathe in many different ways. So as humans, right, we breathe oxygen. We do not live without oxygen, right? And so I kind of have the humans and the animals and the plants kind of at the top. So we call that aerobic respiration. So it's kind of a process where we get, use an energy source like an organic compound, convert it to carbon dioxide, and we use oxygen to breathe, right? Producing water. So we call that aerobic respiration. And there's lots of microbes that do aerobic respiration just like us. But there's also a whole lot of microorganisms that can use other different compounds to breathe or to respire. So we call that anaerobic respiration. So they can use different substances like nitrate, ferric iron, sulfate, and CO2, okay? And they can actually couple that to the metabolism of different types of organic compounds. So I'm going to be talking a little bit later on the sulfate reduction here, because this is what we know, a group that's really important for a lot of microbial corrosion. Yeah, so just keep in mind that microbes have these amazing abilities. And I guess the last kind of, background information on microbes I'll talk about is their energy strategy. So as I mentioned earlier, if you have, feed a microbe a substance like glucose, there's energy stored in the bonds of that compound. And as they metabolize it, they capture that energy and convert it into a source of energy that the cells can use to do many things that they have to do. And that's actually called ATP. So a lot of this is a little bit extraneous, but what I do want you to remember is ATP is the energy currency of the cell. So they will, again, be making that, and we can actually use that as a tool for measuring microorganisms in actual field samples. Okay, so that was my high-level snapshot of microbiology. Okay, very, very fast. We teach whole courses, several courses on this topic. But just a quick review. Microbes exist in almost every known natural habitat on Earth, and I guess we could say unnatural habitats or man-made habitats as well. Microbes can respire aerobically or anaerobically. DNA is the blueprint of life. Microbes will extract energy from nutrients by using different redox reactions, and that's stored in chemical bonds. And again, ATP is the main metabolic currency of the cell. And many different environmental factors will influence what kind of microbes grow. And then that biofilm mode of life is very advantageous for microbes. So again, I've highlighted DNA and ATP because you will be this afternoon measuring or working with those types of biomolecules. Okay. Any questions so far? Following along okay? Yeah, okay. Okay, so I'm going to switch gears a little bit now to talking, that's kind of our little fundamentals about microbes. And now we're going to talk a little bit about microbiologically influenced corrosion, or MIC. And I know that you all know a lot about corrosion and a lot about different corrosion mechanisms, probably way more than I know. But as a very simplistic view, as from a microbiologist point of view, we can think about corrosion as being abiotic. So there's no biology involved in that. So it could be due to oxygen, CO2 corrosion, acid corrosion, H2S, concentration cell, et cetera. So we know there's those types of corrosion out there. When we talk about biotic, this is where we're talking generally about microbiologically influenced. So what this means is that microorganisms have some kind of an effect on abiotic conditions, okay? So in reality, when we're thinking about corrosion, it's really a combination. So microorganisms are kind of everywhere. Most corrosion is electrochemical, and somehow microbes are accelerating those electrochemical reactions, okay? So you kind of have to think about, again, not only the microorganisms doing something, but it's kind of accelerating anything that might happen abiologically. Okay, so coming back to this diagram that I showed you right at the beginning, where we know MIC is a type of corrosion that is affected by the presence and or activity of microorganisms. We talked about the fact that we have to have microorganisms, medium, and metals. We can also think about microbial corrosion as being really an electrochemical process that's just part of the environment, because microbes are kind of everywhere and they're probably doing some influencing on those electrochemical processes. I'm going to talk to you about some single mechanisms that, as microbiologists, have kind of worked out some ways that microbes are possibly degrading, let's say, a metal. But in reality, MIC is not a single mechanism, okay? And usually this multiple lines of evidence are needed. Torben is going to talk a lot about this in his talk. So it's abbreviated MLOE. And what that means, again, is putting the microorganisms that we find in the context of their chemistry and those metals, okay? Okay, so where does MIC actually occur? Do you guys have any idea about places where MIC might occur? Any sectors or any, have you thought about microbial corrosion at all? Yeah. Yeah, so anywhere where water is, absolutely, yeah. Can you think of any particular examples?
Definitely like pipelines or storage, especially like where there's stagnant flow.
Yeah, absolutely. Pipeline systems where places, water lines where there's lots of stagnant flow, absolutely. Yeah, so it does actually occur in many different sectors. Okay, that's not maybe a surprise. And then Torben's gonna come up and talk about some of these sectors.
So
because
yeah,
he gave these slides to me and I thought they were awesome. He said, I'll talk about them.
we'll let Lisa rest her voice a little bit. So yeah, correctly, these technical systems that we have built to facilitate our water infrastructure, we will see these things. Sewer systems, sewer system is like an interesting place. We have a lot of concrete, and there will be a water wetted area, and then there will be an atmospheric area in these systems. And in the liquid, there is one kind of bacteria that is producing hydrogen sulfide. So they're sitting down here in the sludge or in the biofilm, and they're called sulfate reducing bacteria. So they produce H2S. That H2S goes into the gas phase where we have oxygen. And then there are some creatures up in this part where we have a combination of oxygen and H2S, and they're called the sulfide oxidizing bacteria, a totally other kind of bacteria. And they can actually respire on oxygen, and then they use the energy from the H2S, and then they produce sulfuric acid. And it's the sulfuric acid that degrades the concrete. So it's this interplay between two different kinds of microorganisms. So that's one place, and it's a tremendous problem all over the world, and people haven't found a way to come across this or to solve this. So if you come up with an engineering solution, I would love to hear about it. Drinking water systems. This is a typical drinking water system of an old town in Denmark, 150 years old. When we have pressure release in the system, we often get this reddish stuff in our drinking water. It's not dangerous or anything. It's just a bit annoying. But in these, we have a lot of sulfur-reducing bacteria. Again, anaerobic. I love this picture. This was some excavations that we did together with the municipality. That check, yeah.
Oh, but we have a lot of water.
We talked about that.
Talk on Friday.
Yeah, that's it's an ongoing topic. What we do, we actually change our cast iron, asbestos cement, our PVC that was laid in the 70s. We changed that to high density polyethylene pipe. So that's an ongoing process. But yeah, it's like a zoo inside the water pipes. You even have small higher organisms that are grassing on the bacteria. So these are the kind of things that you don't want to know about. I say it just adds to the protein mix of your drink. Then there's sprinkler systems. So if you look up, you have these nozzles in the roof. And if you take down these plates, you will see pipes. And if there is a fire, you want water to come out. But we've done a lot of research, and this is from a hospital in Norway, where this is the interior of the pipe. And you could imagine if this starts to flow, those nozzles will be clogged and no water will come out. And then the insurance company will come along and say, hey guys, you haven't maintained your water sprinkler system, so we are not going to cover your asses or anything. So
that would be like the stagnant water that
Exactly,
you mentioned,
yeah.
right? So it's
What-
like flushing it routinely just to kind of prevent that debris.
Based on the design, because it's a high pressure header. So that the second that you get that ignition, it's going to come out immediately.
Yeah. So that's right. These new systems are actually inert with an inert gas, so they don't have water in them. And then you have a big tank on the roof. So there is new designs, but this is the old design. Again, it's a massive problem. And then there is our favorite topic, and you'll hear much more about oil and gas. We wrote a whole book, about 600 pages on this. I think you also contribute to that book, Lisa. So you'll hear much more about that. So there was just like 4 different areas that is directly related to our everyday life. So I'll put it over to you again, Lisa.
Yeah, thank you. So just a little bit more, I'm going to kind of give you a couple of examples about how microbes can influence corrosion. And I'm not going to be able to, I don't have enough time to kind of go through all the various mechanisms. I think Torben did provide, and you think have access to this one review article. It's very long, 33 pages, but we wrote a review on MIC a couple of years ago with the idea that, you know, when we study microbial corrosion, again, I've mentioned to you how important it is to have all of these elements, the chemistry, you know, the operating conditions, the microorganisms, but a lot of microbiologists just look at the microorganisms and make conclusions about that. So we wanted to have a review that it's more than just the microorganisms. So if you are interested in this topic, you can read further about some of the other mechanisms I won't be able to talk about today. Okay, so I've shown you this diagram before, and I've mentioned to you that there's a lot of microbes that can kind of respire. They can kind of use these electron acceptors to gain energy, just like we would use oxygen. And the organisms, as I mentioned, that were probably the most well-known culprits of microbial corrosion are the organisms that carry out this process called sulfate reduction. So we call them sulfate reducers. They can be bacteria, they can be archaea. I often call them sulfate reducing microorganisms to be inclusive. They've often been known as sulfate reducing bacteria, but we also know that there's types of archaea, just kind of a different sort of line of the evolutionary tree, but they're still considered microorganisms. So they can take things like organic compounds or even things like hydrogen, and as they gain energy from those compounds, they couple the conversion of sulfate into hydrogen sulfide. So Torben just mentioned a couple of those examples. We also know that methanogens or acetogens can convert, that can contribute to corrosion. And so, yeah, there's kind of a lot of literature that's kind of coming up on this topic as well. So sulfate reducers, again, are the most well-studied culprits, but I should have you know that there's many, many types of microorganisms that can contribute to MIC. It's just that sulfate reducers have been the most well-studied. Okay, so I have a little diagram, cartoon that I drew for my class that I teach on petroleum microbiology, just to kind of demonstrate some of the different mechanisms, how microorganisms are thought, and especially focusing on sulfate reducers, are thought to contribute to corrosion. So this is supposed to just be a piece of metal in water, okay? And so we know that when you put a piece of iron in water, it'll readily give off electrons, okay? So, and this, I'm sure you guys know more about this than I do in particular, but you're going to get the dissolution of that salt, of that iron element into ferrous, iron, and electrons, okay? And then in that, under anaerobic conditions, you're also going to have the reaction here on your right, which often just kind of happens chemically as well. Okay, so those kind of reactions can happen when you just put a piece of iron into water. But now you can bring some microbes into the system. Okay, so on the top here, I have some sulfate reducing microorganisms. And so they've come along and they've found some, oil organics or some hydrogen, and they're able to convert it or metabolize it to CO2 using sulfate to breathe as their electron acceptor, converting it to hydrogen sulfide. Okay, and so what happens then in this type of scenario is that hydrogen sulfide can chemically react with that iron 2 just from the dissolution of metal to form iron sulfide. Okay, so it's a black precipitate that can then form on the surface of that metal. So this type of microbial corrosion is actually, we refer to it as chemical MIC or CMIC, also known as metabolite MIC. And the reason it's called metabolite MIC is the idea is that the microorganism produces through its metabolism some sort of metabolite or end product that can then chemically react with some component of that metal, okay? So we have this external electron donor. Okay, so that iron sulfide chemically forms from sulfide and iron 2, okay? The reason that is a problem is that in the natural world, iron sulfide is usually formed kind of in a non-uniform layer. It kind of sets up a corrosion cell with the metal, and that will actually accelerate corrosion. So the fact that this kind of product is generated because of that sulfide being produced from those sulfate reducers, you can get a lot. It's quite an aggressive type of corrosion. But we also know that iron sulfide can actually passivate metal, so it actually can form a protective layer on the surface of that metal as well. So this is often seen in the laboratory when we have an organic electron donor present, and it's usually, we get very low corrosion rates. Okay, so but in real life, if you have sulfate reducers in your system, you kind of get this patchy formation of iron sulfide that is very, very corrosive to the system. Okay, so we've talked a little bit about that CMIC scenario. And there's a second mechanism that was discovered relatively recently, like maybe 10 or 12 years ago, that was initially referred to as EMIC or electrical MIC. Okay, that was kind of the term that was coined. And so in this case, hopefully you can see the difference here, but in EMIC, the microorganisms are somehow directly using the electrons from the metal, right? So when that iron sort of dissolves in water into iron 2 and 2 electrons, those organisms can use those electrons directly from the metal to drive that sulfate reduction to HS, or hydrogen sulfide. And then you're still going to get the formation of iron sulfide as well. So this is a process known as EMIC. And with EMIC, it can actually be direct or indirect. So there's been a lot of debate as to, can microbes actually extract electrons from metal, right? Like there's probably something more going on. And so different people have proposed different types of mechanisms. And this is where it's really important to kind of understand your microbial cell a little bit, because maybe you can start to attribute, how are the microbes actually doing this? So one possibility is that they have, I remember I was talking to you about those lipid membranes that surrounds all those microbial cells. There's some proteins in there called cytochromes, and they're actually function in redox reactions. So it's very possible that somehow those outer membrane cytochromes are somehow capturing those electrons from the metal. So that's one hypothesis in which this occurs. A second hypothesis is that hydrogen is being generated and somehow capturing those electrons. So hydrogen generation from the cell is being converted into 2H+. That's then capturing those electrons from that iron dissolution. And or there's a third mechanism where the microbes are kind of producing some sort of chemical or some sort of chemical that can then uptake those electrons, returning them back to the cell. So again, kind of fundamentals of microbiology can really help interpret how these organisms might be directly taking up some of those electrons from the metal. So this is just kind of these are some figures from that paper I suggested you take a look at if you're interested in microbial corrosion, just to kind of summarize some of the different, CMIC versus EMIC. So in CMIC, or metabolite MIC, we have that external electron donor. Sulfide produced from the activity of those sulfate reducers react with iron 2 to make that corrosive FES. And the rates typically have been reported to be about 0.4 millimeters per year on sort of metal pipelines. In terms of electrical MIC, again, it can be direct somehow or indirect. The metal itself is the electron donor, supplying those electrons to sulfate reduction. And so far, a lot of the studies, at least in the laboratory, have shown that this type of mechanism was way more corrosive, so about up to 0.9 millimeters per year. So, these are just some sort of mechanisms that have been, studied in the laboratory setting. And I just wanted to show you kind of one example of an experiment we did quite a long time ago. We were looking at sort of corrosion rates by an EMIC organism, an EMIC sulfate reducer, so we expected the corrosion rates to be higher, versus a CMIC organism where we expected the corrosion rates to be lower. So on this graph, we have average corrosion rates, and each of these little names at the bottom are just different cultures. So these kind of blue bars that you're seeing up here, so these are just some different corrosion rates that we obtained from our EMIC organism, okay? So this is a type of sulfate reducer. It's called desulfovibrio ferrophilus. We fed it, iron was the electron donor, but we also added formate, okay? So when we had another organic carbon present, not just the metal, we had slightly lower corrosion rates. But here, iron was the sole electron donor, right? So we had higher corrosion rates. So that kind of really suggested that somehow this organism is taking those electrons right off the metal and kind of facilitating a faster reaction from the metal. Okay, and that's just our sterile control. So whenever we do microbial corrosion experiments, we also have sterile controls where we have no microbes in the system. So we're always comparing, like abiotic corrosion to when we add microbes into the system. What we always want to see is higher rates when we have microbes present. Okay, so that's kind of what we did this with little beads. We're going to tell you about a little bit later, so they look a little bit kind of shiny and clear. But when we took beads out of the bottles with the EMIC organism, you can see that there's a lot more pitting and stuff happening on that metal. Okay, so again, suggesting that those microbes are eating up that metal, using it as an electron donor. And then kind of here on the bottom, This is kind of the corrosion rates we got with an organism that is not known to use the metal directly as that electron donor, a different type of desulfo Vibrio. So you can see that the corrosion rates are a lot lower. And then when we take a look at the beads, we see that they're nice and shiny, just like the controls, right? So you can actually see some of the differences in the types of organisms, the severity of sort of the emic organisms versus the semic organisms. Okay, so I'm going to wrap up here in just a moment, but I just wanted to talk a little bit about MIC research. So we know a lot, or we think we have a really good understanding of a lot of, the different types of ways that microbes can corrode metal. We can set up experiments with single species, like I just showed you, well-controlled, clean systems. And here we can make fundamental scientific discoveries about MIC, but it's not realistic, right? This doesn't happen in real life, where we have probably thousands of different types of microbes in a system. But it's nice and clean, and it allows us to kind of get some, discover some principles. We can maybe then move to something where a little bit, maybe a little bit less, less pure, a little more complicated, still very controlled, still a very clean system. But maybe we can get a better understanding when we have, maybe 5 or 6 microbes in our system. But in the real world, we have no control over what microbes are there. And this is the scenarios where we have to deal with when we're thinking about microbial corrosion and trying to figure out if it's happening in the real world. So it's very poorly controlled, but very realistic. So messy. All right. So we have to kind of develop these tools to, that maybe we've discovered through this clean system in order to figure out what's going on in that messy system. Okay. Okay. And I'll talk a little bit about some of those tools that we can use later. And I think Torben will be talking a little bit about that as well. So just really overall summary here, there's many possible mechanisms by which MIC might occur, and many different types of microbes can be involved. Microbes will generally accelerate the kinetics of an abiotic reaction. And we have to think about microbes as just being part of a natural environment. So again, we can do a lot of really clean and controlled experiments in the laboratory, but in the natural world, it's going to be quite messy. So even though I've shown you some kind of single proposed mechanisms, MIC is not really, in reality, a single mechanism. It's a collective term for a variety of mechanisms through which microbes alter the kinetics of corrosion reactions by their presence or their activity. So it's not super straightforward. And we do have to, again, have that multiple lines of evidence approach where we're looking at the microbes in the context of the chemistry or the media, and in the context of the metals or the materials that they are interacting with. Yeah, so that's the end of my high-level introduction to microbes and corrosion. Yeah. Any questions at all?
Yeah. We have one question over here. How could you distinguish the MIC from other potential kinds of corrosion mechanisms? And I think it's in the means of measuring coupon results. I think that's something we will cover, like with the multiple line of evidence.
That's a really great question. And that's where we have to get that multiple lines of evidence happening. If the only thing that changes in an experiment is that we're adding the microbes, it's probably due to something the microbes are doing as well. But we can look at the metals, we can look at the microorganisms. I don't do, I think there's people that do. You can kind of look at surface, right? You can do some microscopy to look at surfaces to kind of get an understanding of what might be happening chemically versus, when you have microbes in the system as well. Yeah.
I guess you're trying to measure general corrosion, but what if in those low corrosion rate ones, you look at the cross section, you have things like SEC or like, obviously these aren't stretched, but like you have localized corrosion or pitting potentially, because now you're occluding regions, you have bare spots that are available. Can you?
Yeah, absolutely. There can still be low general corrosion with some pits. Absolutely. A lot of our experiments, we don't see a lot of high general corrosion rates, but when you look at the metal or the beads, there's a lot of pits. It's just not captured yet. So there's definitely some happening there. Yeah. Yeah.
This is a follow-up question because so it's quite easy to know if the microbes you put in matter, right? So because you have the sterile control, you feed them in different ways, and you can check if they formed a biofilm. It might also be possible to know if surface contact is required, because you can You can look where is the biofilm, you can maybe separate them. But then when it comes to understanding which exactly mechanism, this microbe, this is really hard because there are so many factors that change in your experiment, depending on if you use this kind of cells or if you stir or if there's some oxygen or not. And then, so you could think of feeding them in different ways, change the medium, right?
It will absolutely make a difference. Yes.
Yes, exactly.
Yes. Yeah. So that's why it's quite complicated. And especially when you get into like more complicated systems, it's like, how do we know? And so we always have to kind of put those different types of evidence together where the data collectively as a whole suggests that microbes probably contributed to this system. I don't know if it's ever super clear cut, right? Because again, it's an electrochemical process when we're dealing with metals. So maybe that was the main thing. So, I mean, I think I know yeah, so I think you have to kind of, if there's microbes in the system, probably look at them to see if and some of the products that formed. If FES formed, it's probably a microbial product. So they probably did something. But we also have some cases where we don't see FES, but we know there's a lot of microbes there. There's lots of corrosion. So yeah, it's really tough.
And looking at the metal is always good. So
yes, looking at the metal, absolutely. Yeah. And we do that in a very superficial way. Like we maybe can do profilometry or something like that, maybe microscopy, but nothing super detailed.
I mean, like something that would be like really good to demonstrate is you're seeing all this evidence that microbes are creating these biofilms, and there's FES forming on it. There's some way to completely do the same thing without microbes in there. Maybe you have some like polymeric film and you can blow whatever metabolite
Oh yeah.
on top of it, and then you still have the occlusion. There's no microbe there, and then you can actually change the recipe, I guess.
Yeah.
It'd probably be difficult to manufacture or whatever, but I think you could then have a control where it's just the polymeric film. That's not. You can start.
but such a,
Lisa, can you help changing the slides?
Yeah.
which is also not,
How to do that?
Yeah. Yeah, but it's like isolating effects along with it.
it's one of them, but
Need to move on.
conception that
Yeah.
occlusion is the most important. It's
Yes.
actually very rarely.
But it's the baseline, right? You start there and then you start to put the metabolites in there.
So one thing we that's done quite a lot is centrifuging away all the microbes, taking the metabolites, so basically what they excrete, and then re-immersing your coupons with that. So then you can see, is it what
COLS1.
basically poop out that is corroding, or is it something else? So that is a typical control experiment.
Even like a lab on chip or like some very small like microfluidics thing that you can do on your piece of metal to actually like simulate these processes.
The biofilm could be another issue. So
Yeah.
it's a lot of
Prof. We did that one, that we just studied the effect of metabolites. So we grow our bacteria. It was a single microorganism. We took all the excreted metabolites from them by centrifugation, and then we exposed them again to a new sets of coupon. But the corrosion rate was actually lower than sometimes the control. It was showing the effect of the actual bacteria in the medium and on the corrosion itself.
And we also showed in a failed experiment that you need them on the coupons, because the bacteria, they can, depending on what they breathe, they can be on the, sometimes if you have the wrong cell, they grow on the surface to the air because they breathe the air.
It's over there, but not on that.
On your coupons, right? So you can also control that. And only when they come on the coupon, we get
Yeah, and that's why like a lab on chip I think would be smart. If you could figure out some sort of like microfluidics where you can get oxygen or hydrogen and then your metabolites.
They change the matter. They can breathe a lot of things.
Yes,
So
yes.
that's the problem.
But again, just like
Kind of dragging it into, yeah, here we go.
pure science, right? Versus there's so many variables in just using microbiome.
You already shared it on Zoom as well?
Yeah, it looks like it.
All right. Thanks for the discussion. We're eating ourselves a little bit into the,
Technical.
this is the IT. Thank you, Lisa. It's in your hand. All right, so I'll take over the next 45 minutes. Yeah, So yeah, my name is Torben Skovhus. I did a PhD in microbes that lives on marine surfaces, so algae and tunicates, and how bacteria are trying to have a symbiosis with plants and sea creatures to avoid fouling on the surface. And I thought I was going to work with that my entire life. And then I came into a private company and I got the phone from my manager and said, there's this crazy guy from the oil rigs. He wants to know about bacteria in the pipes and they're deteriorating. And I had no idea that that was a thing. So that was pretty hard on starting with industry. And then I worked 25 years with people in the oil industry with their microbiology problems. And I realized there's not a lot of microbiologists around in industry in terms of having that expertise. But I managed to get an employment with DNV, which is like a major engineering company. And they have like 12,000 people employed. And I was the first microbiologist. So I got to work with 12,000 engineers, and I was the manager of a corrosion laboratory. So sometimes I think of myself as more of an engineer than a microbiologist, but actually my professorship is integrating biological sciences together with engineering, so making that bridge. So I did my PhD with Aarhus University and University of New South Wales in Australia, and then I worked with these two companies for 12 years, so that's pure industry. And now I'm back as a docent or professor at VIA. We have a line of climate and supply engineers, so they look at all kind of waters that flow in pipes and also harbors. How do we protect ourselves from too much water? And then I'm supervising a number of students, including Marit from Western, and I'm doing a lot of project management of projects. I don't know if you know where Denmark is, but it's a tiny country on top of Germany. Germany is right here, and we are here in the center. We have a lot of programs. We have 10 centers of applied research, and it's the second largest educational institution in Denmark. So, as I've traveled all the way from my home country till here, I have a little question for you guys. How many countries does Canada share a land border with? And I see 2 fingers. And which countries are that?
That's in Denmark.
It's actually Greenland. And you know about Greenland, which is part of the Danish kingdom. There's a lot of controversy about people from the south that wants to purchase or take over Greenland. But this has been a dispute for 50 years. And it's kind of hilarious because the Canadian Navy came out there and they put a bottle of Canadian whiskey at the flag, and then they changed the flag to the Canadian flag. Then the Danish came and they put down the snaps that drank the whiskey, and they put the Danish flag up, and that went on for like 50 years. They call it like the Whiskey War, but it was actually solved. There was even a beer, Hands Across the Water, that was produced in this fate. But it was actually resolved in Ottawa between the nations of Denmark, Greenland, and Canada 4 years ago. So I just thought that was a fun fact if you didn't know. Denmark also now have 2 land borders with Germany and Canada. So we doubled up on that. So this is my agenda. I'm going to start out from an engineering perspective, and then I'm going to get a little bit more into the MIG assessment. In the second half after the break, we will go much more into failure analysis and how to take samples. So this is just spicing up or giving you a little bit of a flavor of what we're going to do in the second half. Like when I came into the industry as a young microbiologist, I realized where I was situated in the hierarchy in the oil business. Do you know who's at the top, like at the real top of the system? Which kind of technical or scientific disciplines are the most valuable in oil and gas mining?
Geologists?
Exactly, because they can find where the stuff is underground. They are the most precious. Next, engineers, they can get it up. Next, like finance, economy. Next, people on the rigs and so on. And then you move down. Actually, a person that's pretty high up on the rig is a chef, the guy that makes the food, the medic. And you know who's at the bottom? Like, that's the microbiologist. They only call the microbiologist when they have a pinhole and they want the bacteria to go away. Otherwise, they don't want the microbiologist. So I was called into so many meetings in my early career, and they looked at me, just kill those bacteria, get rid of them. We don't want them. Yeah, but it's not that easy. Yeah, but it's like we want a yes, no answer. We don't want this wishy-washy. So I learned this the hard way, but I think this is all about communication, being able to discuss scientific matter, and everybody contributes. But it's been an interesting journey. So generally speaking, we know that we need to control corrosion so damages doesn't occur, but it still occurs. We have several examples of that. And actually, a management system is a way that asset owners can actually facilitate effective corrosion controls and thereby reducing risk. This is really not rocket science. It's a system that you put in place so that you know what you're doing, when you're doing it, why you're doing it, at what cost, and so on. So it's a structured process. And it's a management system. And you have many management systems described in ISO, in AMP, and in other systems for risk, quality, integrity, compliance. One of my good friends, Ali Morshed, Iranian corrosion engineer, he put that in his book. It's like corrosion management is a suite of procedures, strategies, and systems designed and intended to maintain asset integrity through preventing or mitigating corrosion throughout the operations phase of the asset life cycle. So you have to think from start to end of the life cycle. So how could this look like? These are the 3 core activities that you're going to learn about. And I often see it a little bit as you going to the doctor and you have some symptoms, you don't know what's wrong with you, you have an itchy throat, there's something. And the doctor gives you like a threat assessment. It's actually giving you a diagnosis. This is the same with the pipelines. You throw all kind of measures at the system to come up with what's the root cause of this problem that we have. And then after the diagnosis, the doctor gives you a prescription of these tablets. Go take them, you should be all good. That's the mitigation. That could be paint, it could be cathodic protection. There's a number of things that you can add to your system to mitigate the threat. All right, then you come back home and the itching starts to stop, but the doctor told you, I want to see you in 2 weeks. So that's the control. That's the monitoring and evaluation. You need to monitor and keep taking your blood pressure or blood samples over and over. And this is, again, it's not rocket science, but people, they mess up this. They think that they know what the root cause is, then they prescribe something that is going to make it worse. So make sure that you know these details is extremely important. Some of you might have seen these, but this is like in companies where you have a company policy of something. It could be, we want to have no oil spills. We have 0 tolerance for oil spills. That could be a policy. Or we don't want to have any leaks from our pipelines. That could be a policy. And then you move down, and here it gets very operational. That's where engineers come in, and they're going to interpret the policy and then put like real guidelines into practice. Hope that makes sense. Let's talk just for a few slides about risk. That's another concept that people often mix up a little bit. So risk consists of 2 components, consequence and probability. So the probability of something happening and the consequence of it. Say that you have, this is an example, you can get the slides if you like later on, but say you have a pipeline on the bottom of the seafloor, and it can have a certain probability of failing. Say that it's 10%. Then you have seawater going in that pipeline and it's in the sea. So the consequence of that seawater coming out into the seawater is kind of negligible. It's like zero. It doesn't happen. That doesn't matter. But if it was crude oil that was going in that same pipeline, the consequence of that leak would be disaster. It would be like 100%, right? So you will have a lot of marine pollution. So it's the same system. The probability is the same, but the consequence is so dramatically different. So people need to think about the use of the word risk when we're talking engineering systems. And I would say biologists are not very good at that. They call everything risk. So this is risk, probability, and consequence. Please promise me you will remember this to the end of your life. It's super important. And you've probably also seen the risk matrix. It's a way to work with this, which is extremely beneficial in businesses. This is a bow tie diagram where you kind of move in from the one side on the likelihood to try to minimize the likelihood of something happening. And then on the other side, if it did happen, the consequence you want to minimize, so you could have like contingency plans and ways to deal with an oil spill and so on. Yeah. So this is a figure from a study that we did with the DNV, and there's a report behind it. But it's like, do we have it? So one thing is that, do we do it? So you might have it, you might have all the procedures and so on, but you're not doing it. And the other thing that you might be doing something, but you don't have the system. So then you're kind of walking around in blind and doing like various stuff. And you want to be the world-class company up here, company C, because these guys, A and B, they either have it, but they don't know like what the structure or vice versa. So that was a really interesting exercise where we went out to a number of companies and interviewing them and making best practice. So let's just go through these three in brief. So start with the threat assessment. So Lisa already talked about biotic and abiotic mechanisms. And we touched a little bit upon acids and oxygen, flow, under deposit corrosion, galvanic effects, and MIC. So these are some of the threats that you need to know about. Contributing factors from it could be the presence of water, even in really low amount, less than 0.5% of water. That's an ocean for the bacteria. Nutrients, if the environment is suitable, temperature-wise, pH, salinity, etc. Lisa had some ranges up on that. Deposits, bacteria, they love sitting in like chunks of sand or deposits or debris that is sedimented out. And then you need to know about the different consortia of microorganisms. And we have mentioned a few here: acetogens, acid producers, sulfur-reducing bacteria, archaea, and methanogens. So also there is some common threats like in produced water handling, seawater injection systems. In seawater injection systems, you get a lot of seawater in that contains sulfate, and that's the sort of respiration component for the sulfur-reducing bacteria. Dead legs, somebody mentioned stagnant areas. Lower threat is like crude. Also areas where you have high salinity, you have less growth. And then if you have really dry natural gas, then there shouldn't be a threat. I have seen those areas where you have condensate coming out from even dry gas, and that's where you get the problem. So let's jump to prevention. And again, as Lisa said, we do a whole course, like a whole semester course, and mitigation and prevention is like 3 weeks. So trying to skim this down to the bare minimum. But material selection is, of course, a really important one. You want to you want to select the right material for the job. Then there is coatings and linings. You can, put different devices in, like filters, deaerators, scrubbers, and so on. You can change the operating condition, velocity, mechanical cleaning. You have some pictures of pigs right here that you can run through. They come in many, many different shapes and forms. One oil guy, he called me once and he said if I wanted to go with him to the pig farm, and I had no idea what he was talking about. Apparently we were going out to purchase pigs. So do you want to go to the pig farm? Okay. And then chemicals, corrosion inhibitors, biocides, and other process chemicals. So these are all ways. Cathodic protection, I haven't mentioned, that's another one. So let's jump to the last one here, monitoring. So there's three main purposes of monitoring: to identify if the mitigation is actually working. That's always a good thing. And to identify when conditions change. Then you might need to have a new threat assessment. One example could be that for a long time, the oil operators, they have been sourcing seawater through a pipeline. And then from one day to the other, they changed it from crude to seawater or vice versa. So the whole setup actually changes. You need to do a new threat assessment. Another example, I think I have a case on that later. Oil operator, they produce a lot of water while they're actually producing the oil, and they need to get rid of that water. And you are out at open sea, so what do? You have to rinse it, and then you can dump it into the ocean if it's clean enough. But some guy, he was brilliant, and he said, "Oh, let's re-inject it into the reservoir." Yeah, everybody said that's a great idea. And if there had been a microbiologist in the room, or at least a chemist, there would be like a red flag. You can start mixing two types of waters, one with hydrocarbons, nutrient-rich, produced water that comes from down the reservoir, hot, and then seawater that contains sulfate, which is, I said earlier, the breathing material for sulfur-reducing bacteria. It's like a cocktail of disaster. And there's been many reasons for that. So start mixing waters, alarm. That was one of the first phone calls I got. Come help us. We start mixing the water. Now we have a pinhole leak. And then to learn from incidents and failures. I'll come back to failure analysis later. So this figure you will see in a number of different forms and shapes, but it's extremely important that we take in all these types of evidence when we are going to look at microbiology influenced corrosion. And back to the question over there is not one number that tells you. There's really not one number. It's a little bit like a courtroom. You're building a case, and when you have enough evidence, then you could potentially say that with this probability, it's MIC. So materials and corrosion, chemical environment, the physical and operating conditions, and the microbiological status of the system. These are needed. And that's what we call the multiple line of evidence. So just to recap on the corrosion management core activities: diagnosis, root cause, mechanisms, methods. That's kind of in the first one. There's really no limitations to what you can throw. You can use your syntachron, or you can use your highly advanced AFM. You can use whatever method in this one if it's feasible. Then jumping over to these things that we can add to our system to prevent MIC, and then the monitoring program. So I'll jump a little bit over to some insights on the MIC assessment. Yeah, question?
You mentioned like a lot of people kind of fall in the trap in like trying to change the problem without actually identifying with it, like what it actually is. So maybe, for example, I was at Goodyear, a tire plant, working on internship, and we had a really smart engineering technician, and we had this particular area on the extruder where there's a slurry sort of thing or some mixture to actually cool down the rubber, and then there's like a collection pond. and they're having issue with like pH monitoring and then like trying to actually get it to stay a stable pH.
Mhm.
When, and I don't think like through the whole thing they ever thought about like MIC or anything or like any sort of microbiome, because in a tire plant you have H2S that ruins the lights and everything, and then it's not the cleanest environment because it's just, again, rubber, tire, carbon black, all this kind of stuff coming from all over the world too. So like when in this assessment do you actually like, can you identify that your solution is actually making it worse?
It's a good, like, were there any water streams, like wetted areas?
It's all self-contained in the same loop, but it's a really long track.
Hmm.
And then like you have corrosion and whatever along the whole thing. And then again, this was like 5-6 years ago, so I don't remember the whole detail. And it's just, but this was like the one problem he could not handle, and like the smartest guy I think I've ever met.
I've seen a number of places. I come out to a factory, they produce plastic. So, you know, I'll just take, you know, the cover of your phone here, like plastic items, and you mold them, and then you inject the plastic, and then you bring them back again, and it spits out like 1 million of these plastic covers. And the box, the metal box, they have O-rings, and it's cool, and there's water, and they were not using really clean water. They were using recycled water that was so filthy. So they actually contaminated all their malting boxes. So they had millions and millions and millions of euros or dollar worth these, and they were all infected around the O-rings, and like water was coming out, and I was like, oh, what? And I just took one sample from the source water that they were recirculating, and it was so filthy, like, so.
It's crazy too, because like what they were doing, corrosion or pH monitoring system, where like it was supposed to like keep the pH at a certain point, and then they would put the inhibitor in or whatever to change the pH, and then it would just fly off the wall like a couple hours later. And
there's something happening there that shouldn't be. Yes, yeah. All right. So this is one of my favorite figures of where MIC happens, for instance, in the oil and gas infrastructure. And whenever you see like a little red dot here, you will have the influence of microorganisms. Either in the reservoir, they will do souring, so they will add H2S to your product, so it's getting harder to sell. Or in the helicopter fuel tank, you can have some fungi that's degrading the fuel. If anybody has a boat, you will know that you need to clean out your fuel if you store your boat for the winter, because that's a breeding ground for bacteria and fungi. And then there's also the green dots. I think that's how microorganisms, they can actually help us in the green transition, but that's another course, let's say that. But that was one of the first figures that I learned from when I came into the oil and gas business. And I saw the book on Lisa's table when we were preparing this the other day, and the book was from 2005. Yes, so it's 20 years old and it's still valid. Here are some pictures. This is carbon steel, and it's actually part of a pipe. It's 19-inch-ish diameter, and this is 3 centimeters. And here you have another 3 centimeters. And this goop you will see is actually the biofilm. People will call it different things. I will come back to that a little bit later, depending on what kind of discipline they come from. But this is where the magic is happening. And it's just like having a cavity or a hole in your tooth, which is also sulfur-reducing bacteria, by the way. So this is the stuff you don't want to grow too far, because then you need to change the whole pipeline, which is at the bottom of the sea, and it's a big operation. This is looking into some of the valves. We found a lot of the same types of microorganisms there. But I'll come back to this picture in a second. This is the slide you're going to show the management if you want money to do research on MRC, or because they're really interested in not having their product spoiled. It's a safety issue, like hydrogen sulfide is toxic, so you don't want it in the work environment. They can lose the integrity of their platforms, their pipelines. They can have downtime, that they can actually lose the reservoirs or if it's plugged or anything. So that's the management slide. Some audio interference. So we talked about having MIC in drinking water, in sewer systems, sprinkler systems, and this is the oil and gas if we fold that out. And here we have put up some of the usual suspects, arc-care, sulfate reducers, and so on, acid producers. And these are actually from individual research articles where people are looking at produced water, natural gas, gas storage, bitumen gathering, and so on. There is a ton of papers out there on MIC in different systems. I used to say, I'm glad I'm not a PhD student today, because there is 1,000 papers coming out on MIC each year, and you need to know which of those 1,000 you need, these 10, which are the good ones. And that's why you need a good supervisor, because the supervisor will kind of know the groups that are preparing these. But there's a lot of rubbish coming out. I think that's in any field. We just have like a tsunami of stuff that's coming out. But put that to the side. The importance of operating conditions. Lisa discussed that a little bit earlier. There's different kind of media out there: oil, water, gas, brine, condensate, multiphase. These are all the different electrolytes. You have different process facilities, like bacteria in wells, in the topside processes, pipelines, the downstream processing, like towards the refineries. There's different environmental conditions, temperature, nutrients, metal composition, flow regimes, and then the alignment between the design basis and the actual use. So I talked about that a pipeline could be designed for crude oil carrying, and then they suddenly change it because they need to source some seawater, and it was not designed for that. So maybe the material, blah, blah, blah. So these things also need to be in compliance. And then access point for cleaning and biocide dosing. We had that example yesterday in one of the groups where they were actually able to put in a pig receiver and a pig launcher.
There's a question in the chat.
Yep.
The oil and gas itself also should be treated, not only the water.
Yes, yes. So corrosion inhibitor is put into the multiphase line. So that's true. I think this is actually telling the same story as what Lisa talked about earlier, that bacteria has a wide range of opportunities for living. If there's just like the correct niche for the bacteria, they will flourish. There is a very famous, I think, 200-year-old dogma or sentence that says, with bacteria, everything is everywhere. It's the environment that selects. So it's a little bit like we have skin microbes. It's not a problem. But if we get a little cut or something, some of those can actually get into our bloodstream, and then suddenly it becomes a problem. These are some observations that bacteria, they love to sit on surfaces or interfaces. It's actually being in the bulk is actually abnormal. Has anybody seen a similar slide as this ever? So this is a theory that came out 25 years ago. And it actually is very good when you want to explain in engineering system, how does biofilms evolve and how can you treat them? And of course, this is not the way it works in real life. But if you take a brand new toothbrush and start using it, and then this is the development of the biofilm on the spikes on the toothbrush. This was actually where the first research was done out of Montana on toothbrushes. It could be your shower curtain. You wash it, you autoclave it, you have a really nice shower curtain, and then you put it in, and then you monitor the biofilm over time. This is what happens. Also, what happens in your on your tooth. If you don't brush your tooth for a week, you know, you have this funny feeling on. And then this will also happen in your pipeline. So this lonely guy comes along, and there's this brand new pipeline. It just came out of the factory. It settles. Then it starts to colonize. gets more and more, and it meets some other guys. Oh, this is a nice place. Let's have a party. Let's jump onto the surface. Bacteria want to be on the surface for two reasons. There's food and there's sex. Being on a dance floor in a discotheque, it's pretty hard to get your partner right in 3D. It's much easier on 2D. And also the food is on the surface. So that's the two reasons. So these guys see, oh, right, there's a party. Let's jump in. They became more and more, but it's becoming very crowded. So these guys down here, they send signals up to these, go away. So actually bacteria, they produce like hormones, they produce signaling molecules. So they send these guys, it's getting pretty crowded, go away. And then they slough off, and then this guy actually starts all over again when it finds a new niche. And when it gets to this stage, it's really hard to remove the biofilm. You need to get in at these early stages. And this is exponential. So you need to know exactly when you need. So there's a reason why you brush your teeth like morning and evening. There's a reason for that. It's to maintain your teeth. So there's a reason people are putting pigs through the pipelines. It's to maintain the integrity. If anybody's having a boat, this is taking it even further, where you have the conditioning film. So these are the molecules, the food for the bacteria. Then you get the bacteria. Then you get fouling of algae and so on. You have macrofouling, barnacles. And then if this stays on for a long time, you'll finally get penetration of your boat. So getting back to this picture, depending on where you are, what disciplines you come from, you will call it different things. I even heard an oil company, they call it the black sticky stuff. It was like a company name that everybody knew. When they talk about the black sticky stuff, that was the stuff on the inside of the crude oil pipelines. But it consists primarily of organic material, bacteria, archaea, but also inorganics or anorganics as well. Lisa's going to talk, and we're getting close to the break, a lot on different types of sampling and what you need to be aware of, like after the break. So I'm just going to show a few slides here because sampling is an integrated part of doing a good monitoring scheme. If you cannot get your good samples, you're having a very hard time predicting anything. It looks like a soft ice machine is actually an emulsion. So, you know, mayonnaise is like emulsion, soft ice is as well. So it's blended in oil, gas, and water. So this is what it looks like when it comes out after high pressure. That's not a good sample. When after this, you actually run it from the manifold into high pressure separator, where we vent off the gas, the water, and the oil, and it goes through a number of separation trains. And then we can just go and get water out. Water is a proxy of what's happening on the surface. So it's not really a good sample when it comes to MIC, but it tells you, like, if you have a vessel, something comes in, something goes out, you can start doing mass balance of what is happening. So it's actually a very good measure for mass balance. Do you know what this is? A bit hard to see, but it's a pig receiver. So that's the end station of the pig. And they open up the lid and all kind of goop comes out, and then they take the pig out, and there you can get really nice samples. Say that this pig has been traveling 300 kilometers. You don't know where the sample is from. It's from somewhere in between. But it gives you an indication because they run this pig every second day. So this material has actually been built up. Remember the biofilm, the lonely guy that comes and they start to grow. So what comes out over this 300-kilometer pipeline has actually been, it's freshly new slime. Yeah.
And spread the colony more than cleaning it up.
So the crew is there anyway. So this is just like getting rid of the gunk and the stuff that is in the pipeline.
Yeah, but if the biofilm is at the beginning of the pipe, and then this is supposed to clean it up, and then you just push it across the whole pipe, then it's like sprinkling it everywhere, right?
Yeah, but you want to avoid getting to a point where this happens. So you just want to get rid of all this because that's what's triggering the penetration. So you're going to get rid of what is there. But as MIC is a localized phenomena, these samples are not necessarily the best, but they will give you an indication that something is happening, and you can start to trend it to see if like the levels are going up or going down. And this is it's pretty hard to see, but it's like a coin. It's like a corrosion probe that you can put in flush mount in the stream, and then you can retrieve it after a number of months. And we're going to mimic that in the lab. Lisa's going to talk more about that. So we can actually, from that, measure a lot of the surface deposits. We can use all kind of methods, chemical, biological, physical methods on looking at the stuff that sticks to the surface. We can rinse it off, and we can take nice pictures of the pitting, and we can do DNA sequencing and so on. So it's a really precious type of material or sample, but it's, again, you need to know where to put it in. So you can't put it in everywhere. So just the liquid is good for trending or hotspots, maybe where to focus biocide use. Solids, there is a direct link to MIC. It's good for failure investigations. And then these removable surfaces are good for monitoring program, picking up that you have some pitting going on, and it's you can look at general corrosion, you can look at MIC. So just want to finish up with 2 slides here. This is back in 2013. I was called into an American oil company, and they spent a lot of money on mitigating corrosion and MIC by chemical means. So the company hired other companies, and their contract was to keep the corrosion in control. They also had another company that were looking at the risk-based inspection. So we talked about that with Monica the other day. So they're spending a lot of money on doing inspection programs. And then they hired myself and my company to look at sort of the chemical engineering aspects and microbiology aspects. And we had a very low budget. But actually, all these 3 came up with the same message, the same story. So we said, all right, let's merge I think we were not good friends with the companies here and here because they lost business. But we actually made like a corrosion and integrity management program and tried to funnel all the data into the same room so that people could look at it. Because these were sitting in different departments and they were not talking to each other. You won't believe it. But actually getting all the data on the table and you could see it from different angles, it was an eye-opener for me and the company. And this was actually the starting point of working on this document, which is the recommended practice. It came out 5 years ago, and there is a 10-page annex. So these are the kind of documents that engineers, they look at in companies. And this is about risk-based inspection of offshore topside static mechanical equipment. So everything that's on the platform, not downhole or subsurface or anything. But we actually managed, with a student that I supervised from University of Alberta, to write a 10-page annex in this document, which is like 200 pages, on MIC and what you should be aware of. And he came up with this diagram. I'm not going to go into details, but it's actually a way to get a probability of failure, like a number that the engineers can work with. You remember a risk, consequence, and probability. And this is actually a way to go through a number of things here: temperature, water, surface chemistry, chemical species, blah, blah, blah. And then you can actually come up with a number. So you can see, am I on the good side, or am I totally screwed in terms of MIC? So this gives the engineers a protocol, a way to work. So that's a bit instrumental that we actually got to this point. And then you will see this one more time, and I'm up for time in a second. but that's the multiple line of evidence that we're just going to discuss much more. And yeah, these are the chemical, the physical, the materials, and the microbiology. So I think I will stop here. And are there any questions?
Questions because
Okay.
One follow-up question from the previous one, for the oil and gas, if those should also receive biocides, not only corrosion inhibitors. Next question, you see that?
Yeah, on the biocide part, many oil companies, they purchase the biocide as a sort of insurance. We've done something. And if it's actually working or not, that's the big question. But you really need to monitor the corrosion. You're not so much interested in if there's bacteria or not. You're interested in the corrosion and how that's affected when using biocide. It's a little bit like you take these tablets, but you keep having the disease. Maybe